Exploring plant parts, we will explore the anthers, filaments, stigma, style, ovary, ovules, and the pistil. The anther is a crucial component of the male reproductive organ in flowering plants, serving a vital role in the intricate process of reproduction. It is responsible for producing and releasing pollen grains, which facilitate fertilization and enhance genetic diversity among plant species. Located at the tip of the stamen, the anther plays a key role in plant reproduction by ensuring the successful transfer of male gametes to the female reproductive structures. This transfer is not only essential for fertilization but also contributes to the continuation of plant species by enabling cross-pollination, which allows for a mix of genetic material from different plants. The anther undergoes several developmental stages, starting from a simple tissue to a mature structure filled with pollen sacs, each containing numerous pollen grains ready for dispersal. Moreover, the timing of pollen release is finely tuned to coincide with the receptivity of the female ovules, further optimizing the chances of successful reproduction and the survival of the next generation of plants. The filament is a vital structural component of the stamen, the male reproductive organ of flowering plants. Its primary function is to support the anther, positioning it optimally for effective pollen dispersal and pollination. The filament can vary significantly in length, contributing to the overall morphology of the flower, which is crucial for attracting pollinators. Though often overshadowed by the anther's role in pollen production, the filament plays a crucial role in ensuring reproductive success in angiosperms. By maintaining the anther's elevation above the flower, it enhances the likelihood of successful fertilization by facilitating the transfer of pollen to visiting pollinators or the wind. Overall, the filament's structural integrity and adaptability are essential to the reproductive processes that contribute to the diversity and proliferation of flowering plants in various ecosystems. The stigma is the receptive surface of the female reproductive organ in flowering plants, playing a crucial role in pollination and fertilization. As the uppermost part of the pistil, the stigma is specifically adapted to capture and recognize pollen grains, ensuring successful reproduction and the continuation of the plant species. This vital structure not only serves as a landing platform for pollen but also employs a variety of mechanisms to assess pollen viability and compatibility. Its unique structural and biochemical characteristics, which can vary significantly across different species, determine pollen compatibility, facilitating species-specific fertilization. Additionally, the stigma can respond to environmental stimuli and hormonal signals, further influencing its interaction with pollinators and subsequent fertilization processes. Through these intricate functions, the stigma embodies a remarkable evolutionary adaptation, demonstrating the complex interplay between morphology and reproductive success in the plant kingdom. The style is a crucial component of the pistil, the female reproductive organ in flowering plant. It serves as a conduit between the stigma and the ovary, facilitating pollen tube growth and ensuring successful fertilization. During the pollination process, pollen grains land on the stigma and begin to germinate, with the style providing a protected pathway for the pollen tubes to travel down towards the ovary. The structural and biochemical properties of the style play a significant role in determining reproductive success and genetic compatibility in plant. These properties not only influence how pollen tubes grow and navigate through the style, but also affect the selective interactions between the pollen and the ovule, which can lead to successful fertilization or the rejection of incompatible pollen. Furthermore, variations in the length and thickness of the style among different species can also impact pollinator behavior, thereby enhancing or limiting reproductive opportunities. As such, understanding the intricate functions of the style is vital for comprehending the complex dynamics of plant reproduction and evolution. The ovary is a fundamental structure in the reproductive system of flowering plants, housing the ovules that develop into seeds upon fertilization. This essential organ is often situated at the base of the pistil, which is composed of the stigma, style, and ovary, collectively facilitating the process of sexual reproduction. The ovary not only plays a pivotal role in the production of seeds, but also ensures the continuation of plant species through the intricate processes of pollination and fertilization. After successful fertilization, the ovary undergoes significant transformation to develop into a fruit, which serves multiple purposes, including protecting the seeds and aiding in their dispersal. This transformation is crucial as it enhances the reproductive success of the plant, allowing for the propagation of future generations. Understanding the complex structure and diverse functions of the ovary is key to plant biology and agricultural science, as it provides insights into plant development, breeding practices, and the cultivation of fruit-bearing species, ultimately impacting food security and ecosystem health. Ovules are essential structures in the reproductive system of seed plants, serving as the primary site of fertilization and ultimately developing into seeds after successful pollination. Found within the ovary of a flower, each ovule contains the female gametophyte and plays a crucial role in plant reproduction, genetic inheritance, and species survival. These tiny structures are not only responsible for producing the next generation of plants, but they also provide a vital link in the complex processes of fertilization and seed development that ensures genetic diversity. Understanding their intricate structure and varied functions is fundamental in botany, agriculture, and plant breeding, as it allows scientists and farmers to optimize crop yields and develop new varieties that can withstand environmental challenges, enhance food security, and support ecological balance. The study of ovules also sheds light on evolutionary adaptations and the relationship between different plant species, contributing to our broader understanding of plant biology and ecology. The carpel, also known as the pistil in some classifications, is the female reproductive organ of flowering plants, and it is crucial for the continuation of plant species. It typically comprises three main parts: the stigma, which serves as the receptive surface for pollen, the style, a tube-like structure that connects the stigma to the ovary, and the ovary itself, which houses the ovules and ultimately develops into fruit after fertilization. These components work in concert to facilitate pollination, allowing pollen grains to adhere to the stigma, which can lead to fertilization when they travel down the style to reach the ovules. Additionally, the carpel not only supports fertilization and seed development, but it also influences the characteristics of the resulting fruit, shaping its size, texture, and nutritional content. Understanding the structure and function of the carpel is essential in botany, agriculture, and horticulture, as it directly affects fruit and seed production, which are vital for both ecosystem balance and human sustenance. Moreover, studying variations in carpel structure across different plant species can provide insights into evolutionary adaptations and agricultural practices aimed at improving crop yield. The reproductive structures of flowering plants, including male and female organs, function together to ensure successful reproduction in a highly coordinated manner. From pollen production and dispersal to ovule fertilization and subsequent seed development, each component plays a critical role in maintaining plant diversity and supporting agricultural productivity. The intricate processes involved in pollination, which often depend on various pollinators such as bees, butterflies, and even wind, highlight the delicate balance within ecosystems. By studying these structures, scientists and farmers can develop improved breeding techniques, enhance crop yields, and support sustainable agriculture, ultimately leading to food security. Moreover, understanding the genetic variations and environmental factors that influence reproductive success can aid in the cultivation of resilient plant varieties capable of withstanding climate change and pests, thus contributing to a more sustainable agricultural future. I hope you learned something interesting. Remember to always stay knowledgeable. Until next time, keep studying.